So we've got these two different types of amino acids. These are the glucogenic amino acids and the ketogenic amino acids. So depending on whether the amino acid is glucogenic or ketogenic, this basically tells us what type of compound the amino acid will be after we remove its amino group. So the glucogenic amino acids are those that are able to be broken down and converted into our citric acid cycle intermediates. So once the amino acid has its NH2 group removed, it will actually either already be a citric acid cycle intermediate or be able to be converted to one. So we call these the glucogenic amino acids because essentially they are entering the citric acid cycle. Whereas the ketogenic amino acids, these are the ones that can be converted to either acetyl-CoA or acetoacetyl-CoA. And these ones can actually be used in not only the citric acid cycle, because the acetyl-CoA can enter the citric acid cycle, but they can also be used for other reactions that acetyl-CoA are used in, like making ketone bodies or even making fatty acids in lipogenesis. So an example of our glucogenic amino acid is glutamate, whereas ketogenic amino acids are things like leucine and lysine. So amino acids that are converted, degraded into pyruvate actually satisfy both the glucogenic and ketogenic criteria. So that means that they can either be used to create these citric acid cycle intermediates or acetyl-CoA, because pyruvate can be incorporated into the citric acid cycle to make the citric acid cycle intermediates, or it can be converted into acetyl-CoA. So in that way, the amino acids that are both ketogenic and glucogenic satisfy the requirements for each of these groups. So to summarize, the glucogenic amino acids are those that are the citric acid cycle intermediates, whereas the ketogenic amino acids are those that are the acetyl-CoA that feed into different processes that acetyl-CoA is used for. We've got a couple listed here. Whereas if the amino acid breaks down into pyruvate once we remove its amino group, then it's both ketogenic and glucogenic because it can be converted into the citric acid cycle intermediates, or it can be converted into acetyl-CoA. So it's both glucogenic and ketogenic. So for example, alanine gets converted into pyruvate once it loses its amino group, so it's both glucogenic and ketogenic. So here we can see a map of which amino acids are glucogenic and which are ketogenic. So everything in blue here are the glucogenic amino acids. So in theory, we can use the oxaloacetate that we can generate from all these glucogenic amino acids to convert back to glucose via our so the oxaloacetate is able to be converted into pyruvate, and the non-carbohydrate source of pyruvate can be used to create glucose via one of our processes we've talked about, gluconeogenesis. So this is actually why these ones in blue get their name. They're glucogenic because they can actually be converted back to glucose. Whereas our ones that are actually get converted into acetyl-CoA, these can actually be used to make ketone bodies as well as feeding into the citric acid cycle. And yes, we can't actually have acetyl-CoA being converted back to pyruvate because we don't have the enzyme to do this. But if you're a amino acid that is both glucogenic and ketogenic, that means you are an amino acid that can be converted into pyruvate, because that pyruvate can get converted into both acetyl-CoA and the citric acid cycle intermediates that eventually lead back to glucose. So to summarize, glucogenic amino acids can be converted into glucose, that is, the carbon skeletons can, and the carbon skeletons of ketogenic amino acids can get converted into ketone bodies or can enter the citric acid cycle as well. And if you're a pyruvate, then you're both ketogenic and glucogenic. So in humans, we have some of our amino acids that can be made by the body. These are our non-essential amino acids, whereas there are others that are essential and the human body can't actually synthesize these. So for this reason, they must be gained from the diet. So the amino acids that are essential for us can actually be created in 7 to 10 step pathways in bacteria and plants. So although we don't have the enzymes to make these essential amino acids, plants actually do. So in humans, the majority of our amino acids are non-essential. So we've got the non-essential amino acids listed here. So we can actually make these. Whereas the essential amino acids on the left-hand side here actually have to be gained from the diet. So there's actually some specific compounds that are used to generate our non-essential amino acids in humans. So 3-phosphoglycerate and pyruvate, which are used in glycolysis. So phosphoglycerate is an intermediate in glycolysis and pyruvate is the ultimate product. Whereas citric acid cycle intermediates, oxaloacetate and alpha-ketoglutarate can also be turned into amino acids by accepting NH2 groups. So all of these become amino acids in transamination reactions where they are accepting an NH2 group. It might also require some other modification in the case of 3-phosphoglycerate, but essentially part of what you have to do to convert these components into amino acids is add the amino group. And tyrosine is an interesting one, because although tyrosine can in theory be produced in the body, we actually need to get another amino acid to do this, and that is our phenylalanine that is essential. So the phenylalanine is essential, and once we have the phenylalanine, we can use that to make the tyrosine using a specific enzyme called phenylalanine hydroxylase. So that actually means the tyrosine itself is non-essential. However, there is a defect that some individuals have where they have a lack of the phenylalanine hydroxylase enzyme, and that actually gives them the genetic disease called phenylketonuria, which we can call PKU. So here's a summary of how we can create some of these non-essential amino acids. So the 3-phosphoglycerate can create things like serine, cysteine, and glycine. Pyruvate can create alanine by simply accepting the NH2. Similarly with the oxaloacetate, to create aspartate just needs the NH2, as does glutamate. But the glutamate and aspartate can actually be further modified to create other amino acids. And as I just showed you, we can have the tyrosine being created from the essential amino acid phenylalanine. So this PKU condition is actually quite interesting because the tyrosine can only be produced in individuals from phenylalanine when we have this phenylalanine hydroxylase enzyme. So individuals without the phenylalanine hydroxylase enzyme actually have the deficiency in being able to make tyrosine because they're not able to convert phenylalanine into tyrosine due to the lack of this enzyme. So essentially we have a break in the pathway here. We're not able to convert phenylalanine into tyrosine due to the lack of this enzyme. So in individuals with phenylketonuria, this actually makes tyrosine an essential amino acid because we're not able to synthesize it ourselves, or the people who have this disease at least, and that means they must gain it from the diet instead. But it's more sinister than that. It's not just the fact that these individuals can't make tyrosine. The phenylalanine actually gets turned into something else. So when the phenylalanine is not being converted into tyrosine, it can actually be converted into another compound called phenylacetate. So we can see that depicted in this picture here. So normally the phenylalanine will be broken down into tyrosine by having the addition of the OH group onto the benzene ring. However, if we don't have our phenylalanine hydroxylase enzyme, there's a block in this pathway. So instead, the phenylalanine actually undergoes a transamination reaction, losing that amino group, being converted to a ketone group, and through the loss of this ketone group, it then forms this toxic compound called phenylacetate. So high levels of phenylacetate are actually associated with mental retardation. So it's very dangerous for individuals that don't have the enzyme to convert phenylalanine into tyrosine to have phenylalanine in the diet. So we need to restrict the amount of phenylalanine in the diet to try and make sure we don't have much of our toxic compound, the phenylacetate, being made, which can actually cause the mental retardation. So you can see here examples of how we make some of these non-essential amino acids. So the pyruvate can undergo transamination to accept the NH2 group to become alanine. Oxaloacetate can accept a amino group to become aspartate, and the alpha-ketoglutarate, which again, all of these on the left-hand side are keto acids, accept the amino group to become glutamate in this case. So alpha-ketoglutarate becomes glutamate. So we saw these interconversions in the transamination reactions that we talked about earlier. So essentially this is how we're able to generate these amino acids in the body, and that's what makes them non-essential amino acids. So a lot of our metabolic reactions that we've talked about to do with protein metabolism do rely on B vitamins as coenzymes. So the B vitamins get converted into these coenzymes. So niacin is used in our NAD plus and also NADH, and we need these for our oxidative deamination. Whereas the pyridoxine is needed for our transamination reactions, and we also have other B vitamins involved with making amino acids through particular biosynthesis reactions. So these are our non-essential amino acids that are made in this way. Alright, so here we have a nice summary of what's going on in amino acid metabolism. So we take off the amino group from the amino acid during our transamination reaction. So we're taking the amino acid from a particular amino group from a particular amino acid, passing it on to alpha-ketoglutarate, and in doing so we make glutamate. And the other half of what we're left with, other than the amino group that is given away, is we now have the keto acids. So we looked at what those keto acids could be. They could be things like pyruvate, which can be used in a variety of different reactions, including gluconeogenesis and citric acid cycle. They could be turned into the acetyl-CoA, or the keto acids could also be our citric acid cycle intermediates. So depending on which of the 20 amino acids we actually started with, that will dictate what the carbon skeleton actually is. So remember, carbon skeleton is another name for our keto acids. So after we've got glutamate from the transamination reaction, we can then use it for two different things, either to make aspartate in another transamination reaction or in a redox reaction to produce our ammonium ion. So both the aspartate and the ammonium ion are needed as inputs into the urea cycle. So the ammonium ion is used to make carbamoyl phosphate, and the aspartate feeds in a bit later. But ultimately in the urea cycle, we're able to generate the urea, which is our nitrogen-rich compound that can actually be removed from the body in the urine. So after producing the urea in the liver through the urea cycle, we take it to the kidney for removal. So hopefully you can understand how these different processes connect now. So these overviews are a really good way of checking whether you've understood exactly what's going on in these sessions, and although the details of these pathways are important, the most important thing to understand is actually the connections between the pathways. So once you understand that, you can add the detail in after. But we do need to understand the fact that we need the transamination reactions to move the amino group onto glutamate, which is then used in other reactions like oxidative deamination to create the ammonium ion, which feeds into the urea cycle. And ultimately, we're creating our urea, which we can remove through the urine. So that completes the puzzle of our metabolism. So we've covered in depth our carbohydrate metabolism and also lipid, and today we've looked at the amino acid metabolism. So you can see that there's plenty of different connections between all of the different types of metabolism. Mostly those connections are around acetyl-CoA, because acetyl-CoA can actually be produced from the breakdown of lipid, carbohydrate, and also amino acids. So that's why we call the acetyl-CoA the central metabolite. So most of the connections from amino acid metabolism to the other branches of metabolism are via these keto acids, or the carbon skeletons. So remember, this is what's left over after we remove the amino group from the amino acid. So some of these feed into the citric acid cycle, some can be pyruvate, and some are acetyl-CoA. So can be converted into acetyl-CoA. And then the amino group is fed into the urea cycle, where we're actually making our fumarate, which also feeds into the citric acid cycle. So got those connections there. So in the next session, we'll actually be looking at some of the major connections between the three different branches of metabolism. So it'll be a really good opportunity to get your head around a few of the different processes that we've looked at, and work out how and where these are connected and what that means in terms of different fuels being used at different times, and when you would use one process over another, these types of things. So I'll see you for our next session.